There are two terms that pretty much, if you understand these terms, it's probably the most important thing to know about blood typing, but I'm going to say right now. If you know these two terms, then you don't have to memorize anything. You'll be able to logically figure out everything else. The first term is antigen, and an antigen is a marker that is on the surface of the cell. There are two types of antigens that are on the surface of the red blood cell, A or B. There's actually a lot of other antigens too, but you would use that more if you were trying to catch a criminal or you were trying to match blood and all of that. For our purposes in healthcare, the two antigens, the two markers that are most important for transfusion purposes is the A and the B antigen. Okay? The second word is antibody. And an antibody is something which attacks a foreign antigen. So pretty much if you see a, you've got all blood cells with the A marker, the A antigen, and you have a different type of blood that comes in there. Say you have B blood that comes there, it's not supposed to be there. So your A red blood cells will make B antibodies to attack the B antigen. It's a lot like if you have like a family reunion or something. You're having a family reunion. Okay, so Nastasia, your family, your last name Faison, right? So you have all the people who, I know that they don't all have the last name, but let's pretend that everybody has last name Faison. Okay, so it's everybody. Then what happens, Anthony Rumen comes to your family reunion. Rumen is not Faison. So it's a foreign marker. It's a foreign antigen, if you will. He's like the foreign antigen coming to the family reunion. So what does Nastasia's family do? They call the police. The police are like the antibodies, okay? And what they do is they round up Anthony, and just like antibodies, they round up the wrong cell, throw him in the paddy wagon, take him downtown. So you got the popo throwing Anthony in jail, in the slammer, right? That's what antibodies would do, is basically act like the police, and they round up foreign antigens and they dispose of them. So that's pretty much what you need to know for blood typing. An antigen and what an antibody is. If you know those two things, the rest is very intuitive. Okay, so essentially our blood types, the name of the blood type that we have is named that because of the antigens that are on the surface of the cell. So type A blood has which antigen? Right, A. B has B. And type AB blood?
There's neither. Or A.
A and B. And O?
Neither.
None. Has neither A nor B. So it's kind of like a little bald head. There's nothing there. I mean, like I said, there really are antigens there, but for our purposes, we're worried about A and B. Okay, so why can we have AB blood? Okay, you probably remember from school or something when you talked about Mendelian genetics, you talked about dominant and recessive genes. And if you had a dominant gene, then the recessive gene wouldn't show up. You had to have two recessive genes, right? Well, in the case of blood type inheritance, there is something known as codominance. So we know that we have chromosomes, 46 or 23 pairs. The pairs are pairs because we get one of them from our mom and one from our dad. In the case of blood typing, if say the paternal gene was an A gene and the maternal gene was B, both of them would be expressed and make the AB blood type. And that's what codominance is. Both genes are dominant at the same time. One is not more dominant than the other. Okay, so unfortunately, I think there is some chalk back here. Like no chalk up here, and I don't have anything to write with. I'm like writing in a little stub. Oh, I thought that was a bug. It's not. That's pretty good. This is good. Thanks. Okay, so now, antibodies. What kind of an antibody would A blood make? B. That's correct, because basically A blood has the A marker, recognizes A blood. If you introduce B blood into that A blood, just like my example with the family reunion, then you're going to produce an antibody to attack that foreign type. So what about B blood? What kind of antibody? A and AB blood. Neither. Why? Because AB blood has both markers, right? And therefore sees nothing as foreign. So there are no antibodies. And what about O? A and B, right? Okay, everybody okay so far?
I'm just wondering, why is it again that type A has B antibodies? I know you explained it. So I don't know, that's it.
Because A blood only recognizes other A blood as normal.
Okay.
If you introduce B blood into A blood, it sees it as foreign and it produces antibodies against B.
Okay, so the type A makes B antibodies because A blood is only recognizes other A blood as normal.
Right. Good. Any other questions?
Would you ask that on my midterm, like why that is?
No, but I could ask you what kind of antibodies that it would make.
Oh, got it.
Okay, so now, the next question is, who can we donate to if we have type A blood? Okay, so I have type A blood, who can I donate my blood to? Definitely A. Anybody else? You could donate to AB also because there's no antibodies there, okay? And what about B blood? You can always donate to the same blood type, right?
Not
So
the B then.
B and AB, same thing. Okay, what about AB blood? Who can they donate to? AB. Can they donate to A? No, because A makes B antibodies. Can they donate to A? No, because A makes B antibodies, right? They donate to O, O makes both A and B, so that wouldn't work either. So only AB blood can they donate to. What about O? Everybody, because it's like a ghost. It doesn't have any antigens, therefore no antibodies will attack it. So it can donate to A, B, AB, and O. And so that's why O is called the universal donor.
Because it can donate to anybody.
Yep. Any other questions? Any questions about this? Alright, so next, who can type A blood receive from? They can receive from, can they receive from A?
Type A blood, you mean?
Yeah, they can receive A blood, sure. Can they receive B? No. Can they receive AB? No. Why? Because A blood makes B antibodies, it's going to attack the B antigen. So it can't receive from there, but it can receive from O. Okay, what about B? Who can they receive from?
B.
And?
O.
Yep, for the same reasons that we just talked about. AB blood, who can they receive from?
A, B, and O.
Can they receive from, let's start with A. Can they receive from A? Why not? What about B? because AB blood doesn't make any antibodies, there's nothing that's gonna attack. So it is known as the universal recipient. And who can O receive from?
Nobody.
Well, somebody.
O.
O, right, only O, because it makes antibodies against everybody else. Okay. So the last, now see, again, if you understand what an antigen is, what an antibody is, you really don't have to memorize this chart. You can just logically figure it out, right?
Maybe.
Well, we'll do some more practice. You'll have some more practice in a little bit. And some practice you can do at home also. I'm going to show you this website. It's really fun. Allele. This is actually the genetic combination that a person can have. Okay, that's what alleles are. They're basically the genes. So if a person is type A blood, what combination of genes could they have? They could have from the dad and the mom, they could have two A genes. We designate blood types with a lowercase I. So they could be IA. That means that they have an A gene from the mom and an A gene from dad. And they could also have one A gene from one parent and one empty gene. Because if the A gene is there, you're gonna have the A antigen. What kind of allele combination could you have for B blood?
IB,
and? Yeah, IB and then an empty gene there, I. But AB blood, we have to have what? Both. You have to have both an A gene and you have to have a B gene. Otherwise, it can't be AB blood. And O, what do we have to have?
O.
Just two empty genes, because if you had an A or a B on the other gene, that would be the type of blood that it would be. Okay, does this make some sense to you?
So AB would be like one gene from either mom or dad and one empty one.
No, AB, you have to have one A gene from one parent and one B gene from the other.
Oh, that's not enough, yeah.
Yeah, you have to have both.
And then two empty genes for O and then.
Yeah, you can't have either an A or a B. You have to have nothing. Otherwise, it would be that blood type.
Okay.
All right, so now what I want to show you, we have like 3 slides, I think, on blood typing. I'm going to ask you, okay, that table, this table, I want you now to stop looking at it. Don't look at it anymore. Because what I'm going to show you next is how to figure out what kind of blood that you have. And it's actually a very simple thing, but people get confused if they try to look at the table or try to figure it out. I'm telling you, don't look at the table. I'm going to show you how you can read a blood typing test. Once you know what your blood type is, then this table is useful to know who they can donate to, who they can receive from. But doing a blood typing test, simply all it is looking for an antigen to react with an antibody. And if you get that reaction, that's the blood type it is. Okay, that you need to keep that in mind. Okay. Let's see here. And then once we do this, I have a little game, and then we can do the activity and answer the questions. Okay. So just a little review here. We already talked about the antigens we have on the surface of the blood cells specify the blood type. Type A blood has A antigens, B antigens, AB has both, and O has neither, as we just described. Now, if a person has A blood okay, which they're designated here with these little triangular looking structures, okay? If we introduce A blood into a person's blood that is B, what happens is that the B blood is going to make the A antibody, as we talked about before, right? So the A antibody, see it has little receptors specific for the shape of that antigen. What it's going to do is it's going to cluster the red blood cells together, and it causes a clumping reaction known as an agglutination reaction. It agglutinates and it clumps together. If we took B blood and we introduce it into A blood type, then we have here some B antibodies being produced by that A blood, and the same thing would happen. That makes sense?
Yeah.
Anybody have any questions? Okay. So now, this next slide is showing us what you're going to see when you do your blood typing test. So I'll describe a little bit here. It's very simple. We have these trays which have 3 wells. There's one that says A, one that says B, and the other that says RH. And what we're going to do is take one person, Ms. Brown, Mr. Green, Mr. Jones, or Mr. Smith. We're going to put about 3 or 4 drops of blood into each well. And then, into each well, we're going to take the antibodies and we're going to put an antibody. So for example, this antibody is RH. I'm going to put about 3 or 4 drops of RH antibody into the RH well. Then I'm going to do the same for the B antibody in the B well. And then the A antibody into the A well. And then you can take these little toothpicks and you can sort of stir it around. Just make sure that you don't cross-contaminate. You want to use one side for one tray and then use the other side for the other little well. If you want, you can also shake it a little bit, that works, but you just want to make sure that you don't, like, splash from one well to the other. So what's going to happen, okay, again, don't look at this table right now. So what they did here, they have a person where they had the blood, they added an antibody into one of 2 wells. So type A blood, we know it's type A blood because with the B antibody, there is no reaction, there's no agglutination. But with the A antibody, we did get agglutination. So it has to be type A blood. Because if you have the A antibody that you added to the blood and it clumps, that means that the A antigen is there. And if the A antigen is there, that's the blood type it is. So, very simply stated, wherever you see a clumping reaction, that's the blood type it is. If you have agglutination over here, like they're showing, okay, here there is an agglutination reaction with the B antibody and none with the A antibody. Therefore, we've had an antigen-antibody reaction with B. That means it's type B blood. And likewise, AB blood, we would get an agglutination reaction with both, meaning that we have both the A and the B antigens on the surface. What do you think you would get with O? You think you'd get agglutination anywhere? None. You would have none. If you did not get an agglutination with the A or the B antibodies, there is nothing on the surface of that cell, and it's O. Once you know your blood type, then you can use this table. But first you need to figure out what your blood type is. Okay, and lastly, RH. So RH is another type of antigen that we have. And it's really, you either have it or you don't. If you don't have it, you're RH negative. If you have it, you're RH positive. So if you're RH positive, you don't make I'm sorry, I did it wrong. If you're Rh positive, you actually make Rh antibodies. Wait a minute. Shoot, I'm doing this wrong again. I've only taught this like 50 times, and now I can't think of it anymore.
It says RH blood would never have RH antibodies. RH plus blood would never have RH antibodies.
Where is it? It's up there.
RH plus blood would never have RH antibodies.
Yeah. That's what I was gonna write initially, and it just looked wrong. Okay, so Rh positive makes no, of course, yes, makes no antibodies. Rh negative blood, however, makes Rh antibodies. Okay, so why is this? Because if somebody's Rh negative, they don't have the antigen. So if you introduce Rh positive blood into somebody with Rh negative blood, and that cell sees that antigen, it sees it as foreign. So it will produce antibodies against the Rh antigen. But Rh positive blood, if you introduce Rh positive blood, it's okay, right? Because it has the antigen too. But if you introduce Rh negative blood, it doesn't matter because there's nothing there. So there's no antibodies for somebody who has Rh positive blood. So what happens is sometimes, if you have a mom, okay, if you have a mother who's pregnant, and if she is Rh negative, regardless of what the father is, they used to test the father too, but there probably were too many Maury Povich situations, so they stopped doing that. Essentially, if mom is Rh negative, then they're going to give her something called Rogam. Rogam is going to prevent the mother's immune system from making Rh antibodies. Now, this is not a problem usually with the first pregnancy. See, if the father happens to be Rh positive, the fetal blood could be Rh positive. So essentially, the mother, during her first pregnancy doesn't usually make a lot of Rh antibodies, but the second pregnancies can be a problem, second, third pregnancy, and so forth. So Rogam is given if she's Rh negative because she can produce antibodies. Do you have any questions about that?
Rhogam's given if she's Rh what?
Negative. She's Rh negative. Okay, if there aren't any questions, we're going to do some practice now. I have this blood typing game. You can go online and just type in Nobel Prize blood typing game, and it'll come up. And it's really a fun game. It's also a little bit of a tutorial in case you get stuck. But, let me see here, I don't think I can make any matter. Okay, so we're going to pick a game. Well, first of all, what we need to do is get this thing up on the screen. There. Okay. So what it's going to have us do, we have 3 patients. We're going to select them, we're going to type their blood, and then we're going to decide which blood we can actually give to the patient. Okay, so the little syringe, it's a little hard to see here. We're going to get some blood, and now we're going to put the blood into 3 tubes. So the first tube has the A antibody in it. Second tube has the B, and the third tube has RH. So now what is the blood type of this person?
Is that RH?
That'd be A negative?
This would be A negative. Why is this negative? Because If you don't have the Rh antibody reacting, that means there's no Rh antigen, so it's negative. So you did get an agglutination with the A antibody, so we have the A antigen. So it's type A negative.
So again, I was typing in, can you explain?
Yeah, because if you get an agglutination with the A antibody, the A antigen is what it's reacted with. And if you have the A antigen, it's type A blood. If you don't have a reaction, like over here, we didn't have anything with B or with the Rh antibody, so that means that B and Rh are not there.
How do you know there's no reaction?
Well, the reaction is the agglutination.
Okay,
See the clumping of the cells over the A2?
yeah.
That's how you know. Okay, so this is A, and then negative. Okay. Next, we need to figure out what blood we can give to this person. So they're A negative, and it's telling us we need two blood bags. So what could we give them? What did you say? A what? AB negative? Well, if you have A blood, they make any antibodies? Okay, but you're right about the A, and you're right about the negative. You can give a person A negative, right? A negative, you can always give them the same blood type, right? So we'll give them that. Let's just see what happens if we give them AB negative. Okay, so we know that's wrong. Okay, what other blood type can we give them?
O.
O negative. Could we give them a positive? No, we couldn't because if we're negative, we're going to make Rh antibodies. So if we gave them positive blood, they would end up, having an agglutination reaction.
So what, you can't give them positive because what?
Because they make Rh antibodies.
They would make RH.
Yeah, if they're negative, you make the antibodies. So therefore, you would not be able.
But if you're positive, you don't make RH antibodies?
If you're positive, you don't. That's correct. Yeah. Okay. All right, so let's look at the next patient here. I remember one time I accidentally poked a person in the eye. But see, I think they fixed that because it was really, accidentally I did that and then they yelled out real loud. But, yeah, it's not like that now. Okay, now what's our blood type?
B positive.
B positive, very good.
Why is it positive then?
Because there's an agglutination reaction
In both?
in RH.
Oh, okay, if there's one in RH, then it's always positive?
Yes,
Okay.
wherever you have an agglutination, that's what your blood type is.
But if there's an agglutination in RH, it's always positive, percent too?
If there's not, it's negative.
Okay, I got it.
Okay, so it says here we have 4 blood bags that we can use, so what blood type can we give them? So B positive, okay, so we can always give them the same blood type, so B positive. Okay, could we give them, oh, see this is what's nice too, because if you just hover over it, it'll tell you what, so this is B positive blood, and it shows that it has the B antigen, and it has the RH antigen, but it doesn't have A, so it helps you. Like I said, along with this game, if you play with it a little bit, there are some tutorial guides as well that are there, in case you get confused. Okay. So B positive, what else? Can we give them B negative?
No.
We could.
No.
We can because if the person's positive, they don't make antibodies again. It's RH, so be positive, be negative, and what else could we give them?
O positive and negative.
Yep, O positive and O negative. Right. Okay, so that one's gonna live. We nearly killed the other one, but that's okay, we got this one. Oh, if a person has an agglutination reaction, the medical way to describe the feeling is an overwhelming sense of doom. Really.
What is that third vial? Like, I can't.
RH.
Okay.
Okay,
RH positive.
Well, it's RH positive. Would you say that the other part was? Well, do we get an agglutination with ARP?
Oh, so it's orange negative?
Well, it's RH positive, but what's the other? It's O. Right? Because we didn't get an agglutination with the A or the B antibody, so you have neither antigen. So O positive. And now what kind of blood can we give them? 2 blood bags. O, we can give them. Can we give them anything else? No, because remember that O blood makes both A and B antibodies. So, O blood can only receive O blood. So we can do O positive and O negative, right? I don't know why this isn't letting me slide it over. Okay. Now, unfortunately, because we tested it out, we're only getting 4 blood drops. You want to try one more time?
No.
Let's try one more time. See if we can get 5 blood drops. Okay. Alright, so here we are. No, I'm not gonna do that. Oh, hey.
Hello.
Okay. So what is the blood type here? A negative. Got it. So we only had agglutination with A. Okay, now what blood can they receive?
A and O.
Yep, A negative and O negative.
But no positive, right?
Right, because people who are Rh negative make antibodies against Rh. So if you gave somebody positive blood and they're Rh negative, they would go after that Rh. So it's gotta be A and O negative. Okay. Next patient. Yeah, so like I said, you can do this at home, you know. It's public and it's free. And it's actually kind of fun. Oh, this is one we already had.
RH negative.
It's which one?
O.
Positive.
Yes. O positive. Yep. Because there's no reaction with A or B, so there's no A or B antigen. Okay, so who do they get then?
O positive and O negative.
Yep, O positive and O negative. Cool. Why is it sticking, but I can't grab the bag with it? Okay, last person. Oh, it's this one.
AB positive.
A and B positive. You have all 3 antigens there. Okay, AB positive. Yeah, so they 4 blood bags.
A.
Universal
B,
receiver,
A,
right? And that's
Yeah.
universal receiver.
AB positive would be the complete and ultimate universal recipient, right? We really could give them any of these, right? Because it's got the A antigen, it's got the B antigen, it doesn't make any antibodies against A or B. It has the RH antigen, it doesn't make antibodies against, so it can take on any of the blood types.
So it'd be AB positive, AB negative, A positive.
A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, O negative,
Okay.
any. What would be the absolute universal donor?
Positive.
Well, if you donated positive blood and somebody had negative blood,
Alright.
right, so O negative would be the absolute universal donor. It could donate to absolutely anybody
Why is it?
because it doesn't have any antigens at all.
Okay, but O positive has antigens.
Yes, RH. And therefore, anything that's negative would make antibodies against it. Okay, right, so that's it for that part. Now, as I mentioned before, we're gonna do this blood taking activity, and it would be good, I think if each person did, like, had one person that they typed, make sure that you're not taking